Recent insights about the developmental origins of schizophrenia, something that's interested me for basically my whole career, is to understand how brains get built from very early in life has something to do with how schizophrenia emerges later in life. We know that many aspects of human behavior seem to have inborn characteristics. Temperament certainly is inborn to a considerable degree very early in life. And it's been of interest to wonder whether there are developmental components to neuropsychiatric disorders, even though most of these are diagnosed during early adult years. We've had a lot of evidence that early developmental adversity plays some role in risk for all behavior disorders. This has come from many studies, really for the last 100 years, looking at obstetrical difficulties, complicated pregnancies, and this has been associated with increased risk for basically all developmental behavior disorders. Attention deficit hyperactivity disorder, schizophrenia, been many studies showing that a complicated delivery or birth is a risk factor, sometimes increases risk 2 to 4-fold over general population risk. It's true of autism, it's true of intellectual disability, it's true of Tourette's syndrome. This was a study that Tom Hyde and I reported over 20 years ago when we looked at identical twins who both had Tourette's syndrome, but they differed in the severity of the tics. And what we showed was that virtually the entire variance in tic severity, these were children who were about 10 years old now, but the entire difference in tic severity could be explained by their APGAR scores at birth. The other big insight that obviously has changed the landscape of psychiatric research in the last 10 years has been the discovery of risk factors in the genome, genetic risk factors. And we now know from these large population-based studies, this is an example of the most recent one reported in schizophrenia last year, that there are many regions of the human genome where variations in genetic sequence seem to be associated with increased risk for schizophrenia. To date, there are over 250 regions of the human genome where there are sequence variants, so-called common polymorphisms in sequence, or SNPs, that account for differences in risk for schizophrenia. And this raises the question, is genetic risk and brain development linked in some sort of a causative chain to the origins of schizophrenia? If this is so, we might expect the genes that are associated with genetic risk for schizophrenia would be abundantly expressed during fetal life. In other words, if they have something to do with building a brain from early in life, they should be expressed in the brain when it's being built early in life. So my group, my colleagues at the Lieber Institute for Brain Development and myself have been looking at this in a number of different ways. And the basic strategy has been to take genes linked to developmental disorders, which include schizophrenia, autism spectrum disorder, intellectual disability, and severe syndromal neurodevelopmental disorders that are associated with epilepsy, severe developmental delay from very early in life. Put together all these genes that come from these big population-based studies, come from sequencing the genome and find mutations that are very seemingly causative and other genetic defects. Put them all together in a gene set and ask whether that set of genes is expressed more abundantly in fetal life than in postnatal life. And as a contrast, we take sets of genes that aren't as clearly linked to developmental time frames, such as neurodegenerative disorders, Alzheimer's disease, frontal lobe dementia, etc. And the question is, are the composite set of genes of these disorders preferentially expressed during fetal life? To do a study like this, you have to have human brain material. You have to have human brain material from prenatal and postnatal life, and at the Lieber Institute for Brain Development at Johns Hopkins in Baltimore, largely through the indefatigable work of Tom Hyde and Joel Kleiman, we have a repository of over 3,000 brains, including over 150 brains from fetal life. We've used a technique for looking at the expression of genes called RNA sequencing, which is pretty much the gold standard for how to measure the abundance of genes expressed in a tissue. RNA sequencing basically is like DNA sequencing. You're just sequencing in a linear order the transcripts of genes expressed in the brain. RNA sequencing has the advantage of being able to identify not just what we think genes look like in the tissue, but also all the transcriptional features that make up the expression of genes in the tissue. This includes what we call exons, what we call expressed regions, transcripts, and these so-called junctions, which are the signatures of novel, previously unrecognized forms of genes. I'm not going to go into much detail, but how we can measure these things is we can measure differences between cases and controls, or we can measure how variation in expression of a gene differs based on the genetic sequence differences, that is, on genotype. So we've done this now in a number of different studies. This was the first study reported a few years ago, where we looked at 36 human brains, normal brains, across 6 different time periods, which included, I don't know if I have is there a way to highlight? No. Okay. Is there it's like a laser pointer or something like that? Whoops. Which one? Oh, okay. No, that's changing. That's changing it. Oh, that's the point. Okay. Okay, good. Got it. So we looked at 6 different time frames: fetal life, immediately postnatal, early childhood, adulthood, midlife, and late life. And we looked at any expressed region of the human genome. We identified what we called, whoops, let me go back. We identified what we called 50,000 differentially expressed regions of the genome that were differentially expressed across these ages. And we validated these in a number of different data sets. These represent signatures of how the human cerebral cortex changes in terms of expression of genes. This was all in the prefrontal cortex of normal brains, changes across the lifespan. So this is a particular region of a gene, a particular exon. This is during fetal life. This is immediately postnatal. This is later. So this is a gene that's very abundantly expressed during fetal life and becomes less abundantly expressed as life goes on. So this is the work of Andrew Jaffe, who's our lead data scientist in the Institute. And what we found was about 50,000 of these differentially expressed regions across development. 40% of these were mapped to previously unrecognized expressed regions of the genome, because much of what we think genes look like in human tissues is not really how they look like in the brain. Most of these were mature RNAs. The majority of these were much more abundant in fetal life than in late life. In many ways, the fetal transcriptome, or the way genes are turned on during fetal life, is sort of a tabula rasa for building the networks and the configurations of a growing adult brain. And these overlapped with many of the kinds of things that you would expect would be important during early fetal life, building neuron signaling, et cetera. We then looked at the basic question of the study. Are the elements of these turned on genes more abundant during fetal life than late life? And basically what we found was all of these different features of how genes are expressed were more abundant during fetal life than late life. In schizophrenia, this was also true of intellectual disability, autism, and syndromal neurodevelopmental disorders. We also found differential expression in Alzheimer's and Parkinson's disease. But here, the whole thing was flipped upside down, because what was different about the time of expression of genes associated with Alzheimer's and Parkinson's disease, they were more abundantly expressed during late life than during fetal life. So both poles of this developmental spectrum were characterized by these associations. I just want to mention, this was a study of one brain region. I can't go into this now. We recently reported earlier this year that as you start to survey other regions of the brain, these kinds of patterns change very dramatically. The brain is not like a liver. You know, you take out half your liver, what you're left with is half a liver. The brain is obviously about information being processed in different regions of the brain in different ways. And when we look at the hippocampal formation, these patterns, even though they show the same relative greater abundance during fetal and postnatal life, the exact characteristics of genes, how they're regulated genetically, does vary across brain regions. All of these data are publicly available to everybody in this audience. If you're interested in looking how any gene changes across development, how any gene is affected by variation in the genetic sequence, and differences in expression of genes between cases with schizophrenia controls, you can go to this website and you can access all of these data. So I strongly suggest that you jot this down. This is a fantastically interesting tool to explore. Now, the other problem, of course, with early brain development, it's not just your genetic sequence that determines what genes get turned on and off. It's also your environmental experience that determines what genes get turned on and off. And environmental experience translates at the genetic level into changing the epigenetic state of the genome. The epigenetic state is defined, the epigenetic state is defined by how the DNA is packaged into these chromatin compartments, and how the sequence is modified by secondary chemical effects such as methylation of DNA. So we did the same study that we did in gene expression, looking at changing in the epigenetic state of the genome across development, and this was done by looking at DNA methylation. I don't want to go into this, but it's exactly the same study. Same 36 brains, replicated in other 36 brains. Instead of now looking at how gene expression changed across these time domains, we looked at how the epigenetic state of the genome changed across these time domains. That is how sensitive the genome appeared to be to environmental influences on it, which determine which genes get turned on and which genes get turned off. You will remember every gene in your body, every cell in your body has the exact same genome, because they all emerge from this fertilized egg with one genome. But a liver cell knows it's not a lung cell, which knows it's not a brain cell, because the epigenetic states of these cells are different, and different genes are turned on and off. The epigenetic state, DNA methylation, packaging of DNA into chromatin, this all determines the availability of the DNA to have the transcriptional machinery see it and turn genes on and off. So what we found was that there was a significant enrichment of these regions of the DNA that are methylated, so-called CPGs, in fetal compared to postnatal life. These were enriched for those regions of the genome that have been associated with genetic risk for schizophrenia. This was not seen in Alzheimer's disease and Parkinson's disease. And when we looked at how DNA methylation changed around the time that the illness is diagnosed in early adult life, a time when there is great environmental complexity and stress, we didn't find any evidence that the DNA methylation changes around that time of life when the diagnosis is made were enriched for these regions of the genome associated with risk. Those were enriched for these fetal-related DNA methylation changes. So finally, we asked another question. We said, okay, we have brains of 200 patients with schizophrenia, hundreds of controls. We can look at the epigenetic state of their genome. Here they are, 40, 45 years of age. Let's look at where they differ. They've been ill for 20, 25 years. Patients with schizophrenia have been exposed to all kinds of adverse environmental experiences, which we know should have some effect on the epigenetic state of their genome, because that's what the environment does. So we said, how do they differ? Do they differ in terms of what happened to them early in life, or they differ in terms of what happened to them when the diagnosis was first made? So we found about 2,000 regions of the genome that were epigenetically different as defined by DNA methylation between schizophrenics and controls. These areas that were different were strongly enriched for the fetal postnatal changes in the epigenetic state, and they were surprisingly statistically significantly depleted for DNA methyl changes around the age of onset. In other words, these people who have been ill for 20, 25 years and have experienced all kinds of environmental complications don't show any consistent, systematic, across individual signatures or tombstones or scars of those adult experiences, but something that happened to them 45 years ago has left an indelible, persistent mark on the epigenetic state of their genome. These results suggest that risk factors for schizophrenia, both genetic and those environmental factors that leave a mark in adult brain, are principally related to early brain development and not to the tumultuous time of clinical diagnosis. So the last point we made in this study of DNA methylation was to show, and we have a much more extensive data set now than this, most of these genetic risk factors that have been associated with schizophrenia are strongly linked to the epigenetic state of the genome, how methylated DNA is. And this suggests, and I think this is a rule for all of psychiatric genetics, schizophrenia risk variants are about how sensitive your genome is to the environment. And that's probably what the critical insight is from these studies. So this is all very interesting biology, but there's clearly a lot missing here. And one of the things that's obviously missing is how genes and the environment interact with each other. So we've been very interested in this question of how do these two things combine in development to influence risk. So just to take you back a second to three things we know for certain about schizophrenia risk. One is we know that there's no schizophrenia gene. There are genes that affect brain development and function, and these genes are from all over the genome. This is what we call a polygenic illness. There's no one gene. There are many different genes combining in different combinations in people. The other thing we know is that the role of early life environment seems to matter in risk. We know this from these studies of obstetric complications. I always like to point out something else we knew that we rarely address in the literature, was that if you look at studies of the concordance rate between dizygotic twins and schizophrenia and healthy siblings in schizophrenia, dizygotic twins are as genetically similar as our siblings from independent pregnancies. But the studies have consistently shown that if you're of a dizygotic twin pair and one of the twins has schizophrenia, there's about a 15 to 20% chance that the other twin will have schizophrenia. For siblings from unrelated pregnancies, who are just as genetically alike as dizygotic twins, rather than the concordance rate between being 15 to 20%, it's 5 to 10%. The difference between dizygotic twins and siblings from independent pregnancies is that dizygotic twins share a uterus. The other thing we know is that schizophrenia, like all developmental behavior disorders, is much more common in males than females. It's about 2 times more common in males than females. This is true of ADHD, intellectual disability, stillbirths, Tourette syndrome, autism. All of these behavior disorders that have a developmental origin are more common in males than females. So how does genetic risk relate to pregnancy complications? Do genetic and viral risk factors that exert their effects during pregnancy act independently, or do they interact to modulate risk? So we were interested in the possibility that we could look at genomic risk, measuring what we call polygene risk scores, and a history of a complicated intrauterine environment to see whether these factors would interact with each other in a non-additive way to increase risk. So polygene risk scores is a summary measure of all the regions of the genome, all the alleles and all the SNPs that identified regions of the genome associated with risk that you can add up in any given individual. And this gives you a sense of genomic risk. Nobody's ill fundamentally because of one SNP here and one SNP there and one p-value here and one p-value there. People are sick presumably because of their genomes and how their genomes relate to their environment. So you can create these polygene risk scores by adding up all the SNPs that were in GWAS significant loci, which we call polygene risk score 1. These were all the loci above the horizontal red line that were called GWAS statistically significant, because they have values of 10 to the minus 8. You can also, there's about 100 SNPs that make up that polygene risk score. You can take the SNPs that were almost significant, 10 to the minus 6, that likely as the sample sizes get bigger become significant. That's about 238 SNPs. And you can keep doing this for increasing number of SNPs. Theoretically, you're accumulating more risk loci. And so that's the strategy we took. And the first thing we did was we looked at a large sample of patients with schizophrenia controls at their polygene risk scores. Each dot is a person. And what this slide shows, which has been shown in many, many studies now, that if you look at a population of people with the diagnosis of schizophrenia and you calculate their polygene risk scores for the polygene risk score 1 set of SNPs, which are the GWAS significant set of SNPs, patients with schizophrenia have higher polygene risk scores than controls. This has been seen now in almost every study that's ever looked at this. This is a reproducible, it's not a huge effect size, it's not a highly biologically significant difference, but it is a valid difference. So then the question we asked was, well, what happens if we divide up the patients and controls based on whether they had a complicated pregnancy? And what we found in our original discovery sample, to our astonishment, was that the difference in polygene risk scores between schizophrenics and controls was only found if the schizophrenics had a complicated pregnancy. And at least in this sample, if they didn't have a complicated pregnancy, there was no difference in this. And if they had a complicated pregnancy, it was a dramatic leap in significance, and there was a significant true non-additive interaction. We had the opportunity to try to replicate this in 2 other samples, including Professor Bertolino's sample from Bari, and both of these other samples showed essentially the same thing, maybe not quite as significant interaction. But again, the polygene risk scores were much greater if patients had a history of obstetric complications than if they didn't. We also looked in 2 independent samples that didn't have controls. This was another, a Japanese sample, which is significant because the polygene risk scores in Japanese samples in general are much less different in schizophrenics and controls than Caucasian European samples. And another German sample. This is the work of 2 postdocs of mine, also from Professor Bertolino's program in Bari, Gianluca Orsini and Giovanna Punzi. And this was the meta-analysis of patients only, again showing these were highly significant differences. Interestingly enough, these differences were only found if you took the polygene risk scores from the most significant genome region loci, 10 to the minus 6 and 10 to the minus 8. All the other ones showed no effect at all, suggesting there was something about the genes in these loci and a history of a complicated pregnancy that seemed to make them speak to each other. How important is severity of an early life complication? ELC is early life complication. If early life complications modulate the impact of genomic risk and vice versa, it would be expected that this interaction would scale with how severe the obstetric complication is. And that's precisely what we found. This is a scale that's been used to measure the severity of obstetric complications and the likelihood that they have an impact on the physiological state of the fetus. And scores above 4 are thought to represent a significant physiological stress to the fetus. Scores below 4, which could just be something like a cord wrapped around the neck, are not thought to have a significant physiological. As you can see, the interaction is only found at the more significant scores at a very, which this is pre-eclampsia. Here there's almost no overlap in polygene risk scores, even though clearly the N is small. This is a meta-analysis of all the case control data. It's a highly significant interaction of this history of a complicated pregnancy and polygene risk scores. But what's much more interesting here is the effect on the prediction of liability. So what you see here is this red mark goes right to this. This is the likelihood that a person is schizophrenic versus control based on polygene risk score and no history of a complicated pregnancy. There's a little bit over a 1.5% probability that you're schizophrenic here. There's almost a 10-fold increase in the prediction of liability if there's a history of an obstetric complication also. And these are graphs just showing how much there's a difference between whether you have a history of complicated pregnancy and not. And again, these effects are only seen for the most significant GWAS loci. All the other loci, where generally we think most of the schizophrenia genes will be found, but the sample sizes have not been large enough yet for these to achieve this special thing called statistical significance. But for some reason, these 2 groups of loci and the genes that map to those regions of the genome seem to be particularly critical for this interaction with obstetric complications. So how do genomic risk and early life complications interact with each other? What is going on in the placenta because the placenta is the critical organ that regulates the development of the fetus. It is a predominantly fetal organ. Many people think the placenta comes from the mother's genome, but that's actually not true. The placenta comes from the fetal genome, and it is the critical organ in mediating the mother's bloodstream and a fetus bloodstream to nurture and lead to development of the fetus. So we've done RNA sequencing analyses in many placental data sets. And these are some examples of regions of different placentae where we looked at the question, the genes in the loci, in the psychiatric risk loci that interact with obstetric complications, which are the PRS1 and PRS2 loci. We looked at all the genes in those loci, and we asked whether those genes were more abundantly expressed in these compartments of the placenta compared to genes in all the other schizophrenia loci which don't interact with obstetric complications. And these are highly more abundantly expressed than are all the other schizophrenia genes. Is the interaction of genomic risk and early life complications dependent on loci with genes dynamically regulated in the placenta? I didn't show this slide, but we also looked at placenta from complicated pregnancies, from preeclampsia, from intrauterine growth restriction, and we found again that the genes in the PRS1 and the PRS2 loci, those loci that interact with obstetric complications, genes in those loci were dynamically regulated in placenta from complicated pregnancies. So we can answer this question. We can test this directly by fractionating polygene risk scores into those loci that have these placental genes that we call plaque polygene risk scores, and those loci that don't have the placental genes called non-plaque polygene loci. And we can take these back to the clinical sample and see whether there's a difference in which of these newly refined polygene risk scores, those about placenta function, those not about placenta function, which one of these predict this interaction with complicated pregnancies. And what you see here is, this is the graphs for the non-plaque polygene risk score gene loci. There is no interaction. The genes in these loci that are not about the placenta have no relationship to the link between genetic risk and obstetrical complications. The placental polygene risk score genes explain this interaction. This interaction between polygene risk scores and complicated pregnancies is because of the biology of the placenta. So how do polygene risk scores influence the biology of the placenta? How do they do this? So animal studies have shown that many stresses during pregnancy that put a physiological stress on the placenta exact an immune response from the placenta. It's very interesting to think about this. The placenta is very much like a tumor growing in a pregnant woman. It is an alien tissue. It comes from the genome of the fetus. It is highly, it grows remarkably from, 2 cells to a pound in 9 months. It is highly invasive. And the mother's immune system is constantly invading the placenta with T cells to try to reject it. And the placenta has to fight off this degree of alien tissue response. So many studies have shown, regardless of what you do to a pregnant animal that puts a stress on the pregnancy and the fetus, there's an immune response initiated by the placenta. So we looked at the expression of 535 immune response genes in the same placentas that we looked at the expression of schizophrenia genes. And what we found was the more the schizophrenia genes are expressed during complicated pregnancies, the more many different immune response genes are also expressed. The schizophrenia genes are monitoring the physiological stress on the placenta during pregnancy. So I also mentioned that one of the other things we know about schizophrenia, like many developmental behavior disorders, is that there's higher frequency of the expression of these in males than females. And as luck would have it, animal studies have shown that outcomes of altered placental functioning on brain development are surprisingly sex-specific, with males much more vulnerable than females to prenatal adversities. Is there a link with the greater incidence of developmental disorders like schizophrenia in males? So what we did now was we looked at the expression of these placental genes in schizophrenia risk loci that are that show this prediction of immune response. And we looked at whether the placenta was from a male or a female. And what we found was all of these genes, in contrast to the non-placental schizophrenia genes, and all the genes in the genome are much more abundantly expressed if the placenta is from a male-female fetus than from a female fetus, which may have something to do with the male risk of schizophrenia being greater. So the last question we've asked, and this is some unpublished data yet, is if this is all playing out very early in fetal life, we might expect that these placental polygene risk score genes, which affect the resilience of the placenta to the stress of pregnancy, might have some influence on early child development. So we've been able to collaborate with John Gilmore at the University of North Carolina, who did this rather extraordinary study of doing MRI scans and tests of cognitive development in infants in the first week of life. They put 240 infants, mostly normal infants, in the MRI scanner. And we asked the question of whether or not these genes, which affect risk for schizophrenia and placenta biology, have some relationship to early development. Here's the bottom line on this. The same polygene risk scores 1 and 2, that predict complicated pregnancies, that have genes in their loci that are dynamically regulated in the placenta from complicated pregnancies, that track the immune state of the placenta. These genes, and only genes in these two loci, predict brain size at birth. There is no relationship between the non-placental genes and brain size at birth. The other remarkable thing is these effects are much greater in males, just as we showed, much greater in males than in females. There's no interaction, probably because the sample size is a little too small, but again, these effects are much greater in males than females. These also predict cognitive development as measured on the Mullins scale at 1 year of age. And again, this is an effect much greater in males than females. So I just want to mention the many people in my years of being in this area of research who had the extraordinary good fortune to work with. And these are the people who contributed to the data I've shown you, many people from Europe. And this is obviously ongoing work in a number of different respects. So the take-home message, I think, is genes associated with developmental disorders are preferentially expressed during fetal life. GWAS loci for schizophrenia, which are the genome-wide association study risk regions of the genome, are enriched for genes showing transcriptional and epigenetic changes related preferentially to early fetal life compared with changes around the time of clinical diagnosis. The most significant genetic variants detected by current GWAS contribute to risk of schizophrenia, at least partly by converging on a developmental trajectory sensitive to intrauterine and perinatal adversity and linked with abnormal placental function. Gene-environment interactions influencing placenta biology may account for the higher incidence of schizophrenia in males compared with females. Genome scores based on schizophrenia risk-associated genes dynamically regulate placenta, predict early brain and cognitive development. And lastly, the holy grail in all of medicine is not treatment, it's prevention. It's much easier to prevent something than it is to treat something. These insights into early brain development, and particularly environment genes interactions in the intrauterine environment, may provide new opportunities for reducing risk. I like to say that when you have in this era of genomic and molecular medicine, the most sophisticated treatment for a complicated pregnancy is bed rest. There have to be more advanced approaches to how to reduce the stress on the developing placenta. This is an image from a paper we published 2 years ago about how to put all of this in some developmental context.